So after the genomes are assembled together, doing annotation is an interesting and very important task. So what we do here is we try to locate some important genes, which are the protein coding genes and their protein products. We try to locate the RNA encoding genes. We recognize the non-coding regions in the genome. We can also predict the functions of the genes, biochemistry and structure of their products. We can explore some literature links. We can also explore the links to the genetic maps where they are located on the chromosomes. We can look into the location of the repeats, location of sequence tag sites, and sequence polymorphisms. Annotation is an important step, which is actually most of the time what we do here is we do comparison. So we find the significant alignments with some proteins of the known functions. And annotations are divided into two types. One is structural annotations and the other one is the functional annotations. So structural annotations are where we actually try to identify certain features which are related to the genes. For example, promoters, terminators, Schindel-Garneau sites, which are the ribosomal binding sites during the protein synthesis. There are some specific patterns of the nucleotides within the genes. We call them as motifs. We look for code transcriptional units, operons in microbes. So in microorganisms, a lot of the genes, they are transcribed together, so we call them as operons. There are two tools which are important or worth mentioning here. One is MAGPIE and the other one is GeneQuiz. So they are designed to assist with genome annotations. MAGPIE stands for Multi-Purpose Automated Genome Project Investigation Environment. Normally it is used for structural annotations, whereas GeneQuiz is good for functional annotations. So here we have an outline that is kind of a workflow how MAGPIE works. So we take some source sequence and we give it to some software program. We call it as MAGPIE daemon. So it takes that sequences which are added to the databases. So it automatically gets them and then it sends that data to some local tools and some remote tools over the internet, and then it explores some specific features or annotation patterns and then it puts them into its feature database. And then later on those results are interpreted and we get the reports. So in this way it's kind of a automated annotation gathering tool. Attaching biological information to the genes is called functional annotations. So what we do here is we attach biological functions, we talk about biochemical functions, we go for gene expression. Normally we go for transcription of the genes as we take it as a gene expression measure, and then regulation and interaction among different genes. There are different classification schemes which are there for this functional classification. So one of them was actually this 8-group classification. So that is to classify the genes and their products into one of these groups, which were like enzymes, transporters, regulators, membranes, structural elements, protein factors, leader peptides. These are the leader peptides actually control transcription and translation, and then we have carriers, transporters. So in this way, scientists, they have seen that 90% of the E. coli genes, they fit into these categories. So their annotations can be explained. There is another scheme, that is enzyme commission numbers or EC numbers. So that was put forward by enzyme commission that was working under the IUBMB, International Union for Biochemistry and Molecular Biology. So they say that enzymes, they are classified on the basis of the reactions they catalyze, and we have a 4-digit scheme which is actually the enzyme commission number. So in which we have first digit we represent here as A. So it's actually this number tells us that this enzyme belongs to one of the 6 class of biochemical reactions. So actually they have 6 classes, so our enzyme can be coming from one of them. Whereas the B is the group of substrate, the thing on which it acts. C tells us about the acceptor molecule, and D is the detail of that biochemical reaction. So for instance, we have an example here, EC 3.4.11.4. So where in the first digit here 3, that means it's hydrolase. So hydrolase is actually the enzyme that uses water to break up something, and 3.4 means it's an hydrolase that acts on peptide bonds. So it's kind of breaking some proteins. 3.4.11 means actually it's an hydrolase that cleaves the amino terminal amino acids on a polypeptide. And the last digit 4, putting everything together. So these are actually the hydrolases that cleave amino terminal amino acid of a tripeptide. So with this enzyme commission scheme, they classified 70% of the E. coli genes into the 70% of the E. coli genes. They shared first 2 classes A and B. So they are kind of catalyzing the same biochemical reactions. There is another scheme that we call it as a 3-group scheme. So divide all those functions into energy related or information related or communication related. So it was found that plants, they devote half of their genome to the energy and metabolism of the energy. Obviously they make food for all of us, whereas animals, they talk a lot. So they spend a lot of their energy in communication. So in the end we conclude that finding genes and their coding regions is an important task in genome annotations. Functional annotations, they correlate genes into different classes of functions.